Hello again, MAP students. If you're at this place in the program, that means that you have successfully completed the green unit. The green unit is a huge unit. It's got the vast majority of the program ended up being in the green unit. So if you've gotten to this point, pat yourselves on the back. You've learned a lot about insulin. And really, at this point, you know enough about insulin structure and function that you can start thinking about your protein story if you'd like. You can continue on in the units if you'd like, but at this point, there's no reason that you can't start figuring out what your modeling project's going to be. So in order for you to do that, though, I want to make sure that all the information and all the knowledge that you've built through this unit makes sense, and so we can try to pull it all together, because like I said, it's a lot. So I want to go through this video to kind of work through it and kind of remind yourselves of everything you've learned to make sure that it all makes sense to you. And if it doesn't, there's a discussion board. You can always ask questions on the discussion board as well. All right, let's get started with the first folder in the green unit, which is basically understanding and modeling basic insulin structure. And in order to do that, you should have used either the insulin mRNA to protein folding kit, or utilized some tubers that you might have and from an amino acid starter kit, and used the instructions that I have in Schoology to try to do it on your own. So if you did that, you should have learned a lot about insulin and insulin structure. So some of the things that hopefully you got from that is that it's clear to you that that insulin is a protein comprised of two chains. Right? So a lot of times we think of proteins as being all one chain. Insulin is unique in that there's two different chains. There is this one in purple here, which is the A chain. That is 21 amino acids long. And I like to think of it as kind of, obviously, proteins don't have an upside and a downside. They kind of, they are in all different forms, but when I hold it like this, I like to think of the A chain as being kind of like an upside down U. So hopefully you can see that there are two alpha helices for the secondary structure of that A chain, and then they're connected by kind of an unstructured connector piece or whatever. So you can see the A chain there. If you turn this tuber model around, I like to think of the B chain, which is 30 amino acids long, as kind of looking like a Z. All right, and then the alpha helix is the middle diagonal part there. So hopefully you can kind of see that there's this N-terminal. This is another term that hopefully you've been learning along the way. The amino terminal, or the N-terminal, and is kind of a beta sheet secondary structure, and the carboxy terminal, or the C terminal, is also kind of a beta sheet. Depending on its structure, sometimes they're more structured than others, and we'll talk about all that later. But that's the basic structure of an insulin protein. You also had to add the disulfide bonds, okay? And you should have learned that there were 3 disulfide bonds in an insulin protein. There is one that is what we would call an intra-chain disulfide bond. So within the A chain, there is a disulfide bond between a cysteine at the A6 position and a cysteine at the A11 position. So disulfide bonds are always bonds that are between 2 cysteine amino acids, and these sulfur groups then are oxidized to form a covalent bond between the two cysteines. So that's the first one. And then there are 2 that are inter-chain disulfide bonds. So we've got one over here that happens to be between A7 and B7 cysteines. And then you got another one on the other end that is between the A20 and the B19 cysteines, okay? So hopefully you modeled those and you can understand then how that kind of holds the protein together. So disulfide bonds, you might not find them in all proteins, but you'll find a number of them in proteins that are secreted to different areas. So they could be secreted into your blood or secreted into the stomach or something like that. Because proteins have to be, secreted proteins have to be in different environments, these disulfide bonds add some stability to keep them from breaking down as they change, as they move from one environment to another. And you'll also find a lot of disulfide bonds in membrane proteins and things like that that need a very tightly maintained structure. Okay, we will talk a little bit more about how that might work as we talk about the processing later on. But those are the main details that you'll want to make sure you understand with the insulin protein. But there is a little bit more to the story. There's an added level of complexity. So I want to kind of touch on this now because this is something we'll be talking about again later. So here are some more insulin proteins. And what you want to know, what you want to understand about these things is that this C-terminal end, the bottom of the Z that we were basically talking about, this part is very hydrophobic. And so when there's a high concentration of insulin, what these insulin proteins want to do is they want to come together along this stretch of amino acids because they'd rather be together with these hydrophobic amino acids than, you know, hanging out in the water. So they come together in a dimer. Not only do they come together in dimers, they actually then will come together with two other dimers and they will make a hexamer. And so we will talk about this more when we talk about processing and storage, because this is the form that insulin is stored in when it's in the insulin granules, when it's ready to be, waiting to be secreted into the blood. So this is something you want to think about. So this is a monomer, this is a dimer, and then this is a hexamer. So I think that's it for that section. Next we'll move on to the synthesis. Okay, so now that we have modeled the structure of insulin, we might want to start asking ourselves, how does this happen? How is this synthesized? So basically what you should have worked on in this unit is understanding the coding of DNA and RNA and how those triplet codons are basically decoded to identify the correct amino acid that needs to be added to the insulin chain as it's being synthesized. But just to back up, let's talk about where is that DNA, okay? The DNA that contains the gene, the insulin gene, is in the nucleus. And what nucleus are we talking about? We're talking about the pancreas. Specifically the beta cell in the pancreas are the specific specialized cells that are going to be making insulin. No other cell in the body does it. It's just these specialized cells. So those cells in their nucleus, they are going to be transcribing the insulin gene, the DNA on the insulin gene, which is located on chromosome 11 in the nucleus of these beta cells, okay? So transcribing means that RNA, a messenger RNA transcript is going to be made, so it's basically a copy is going to be made from the DNA in those cells, in the nucleus of those cells. That transcript then is going to leave the nucleus, enter the cytoplasm, and then that transcript is going to be what we call translated into the insulin peptide, okay? And that is going to happen with a ribosome in the cytoplasm, okay? that ribosome, and we'll talk about this a little bit in more detail when we talk about the processing, but then that ribosome is going to basically decode those triplet codons. So 3 nucleotides are going to be translated into an amino acid one at a time. And that's the activity that you did in this unit, is looking at the codon chart and determining what amino acid is being specified by each triplet codon, okay? So that's what I really want you to understand as we move forward. So now we will talk about how synthesis and processing works in the beta cell. Okay, so now we're going to try to kind of walk through the synthesis to processing stage, and that was in this unit you should have had gone through the notes that we gave you, the activity of trying to kind of work through this, and there are some pictures in there to help it make sense. I thought it would be helpful to try to come up with a model that might help model it as well. So let's try this. It could be a little awkward, but we'll see if we can make some sense of this, okay? So this, in the colors that should have been in your maps and things within the Schoology thing, is the full peptide that is translated in the beta cell by the ribosome, okay? It is what they call pre-proinsulin. So this initial segment here that's colored in yellow, this is called the signal peptide. And what happens is this is going to be the first part of the peptide that's going to be synthesized by the ribosome. And what is going to happen is this is going to be coming out of the ribosome, and it is going to, it's a signal. So there are proteins then that identify this as a protein that needs to be sent to the endoplasmic reticulum. So this then helps the ribosome dock to the endoplasmic reticulum. And then this is, through transport proteins, this is fed into the endoplasmic reticulum as it's being synthesized. As this continues to be added or inserted into the endoplasmic reticulum, this continues to be sent in, and then there's a specific protein that will sense this on the inside of the endoplasmic reticulum and cleave this off. And it kind of goes away. We don't know exactly what happens to this. It may be degraded. It may have other functions. We're learning more and more about these types of things all the time. Okay, so then after the signal peptide has been cleaved, this translated polypeptide starts to form within the endoplasmic reticulum and fold into its final shape. And it does this by the alpha helices and the beta sheets start to form, and this folds onto itself such that the cysteines come into close approximation, and with the help of an enzyme start to form these disulfide bonds, which give it additional stability and structure, as we mentioned before. So then we in this endoplasmic reticulum, we end up with something that looks like this, with this C-peptide that's kind of hanging out here still. But as you can see, it helped kind of pull everything together, and now it's just sitting here. And we know that that's not part of the final protein. Now, we know that in the endoplasmic reticulum then, we not only have these monomers being formed, these hydrophobic interfaces that we were talking about cause the monomers to want to come together in dimers. And then they also start to form hexamers. So what happens is they don't really look quite like this yet. They look something more like this, where you've got a C-peptide that's kind of hanging out on every insulin. So a hexamer is going to have all these different loops hanging out. So these then get transported to the Golgi. And in the Golgi is where the packaging is going to happen. And what turns out is that it's going to package these hexamers into what they call, they're like vesicles, but they call them insulin granules. And these granules have all of the enzymes and other things that they need in order to package these things as hexamers and then crystallize into very high concentrations of insulin. And so what happens is these granules mature over time, and as they mature, the insulin gets to the point where it's ready to be released. So in an immature granule, it'll look like this. But then in these granules, there are specific proteins that cleave this from this area, leaving these two ends. And that causes there to be two insulin chains for each insulin protein as opposed to just one. So through the maturation process, you end up getting hexamers of insulin and a lot of C-peptides hanging around, okay? And hopefully you guys saw in that article, that figure from the article that I put on Schoology, what those insulin granules look like, okay? So there's a lot of these hexamers and a lot of these C-peptides floating around. That article is kind of interesting because it proposes that the C-peptides aren't just, garbage. They aren't something that just gets broken down and gotten rid of. They're saying that there's a lot of research out there now that says that the C-peptide alone has a lot of function. It's protective against apoptosis, it's protective against kidney disease. A lot of the things that that diabetics struggle with, it may be partially because of the lack of the C-peptide, not just the problem with insulin. So that's a really interesting area of research that's happening right now. So now I want to really kind of take a little time to talk about those insulin granules, okay? What are those insulin granules? That figure that I put in Schoology I thought was really kind of cool because it not only shows you what a beta cell looks like in the pancreas, so that's kind of interesting to see, and you can see the insulin granules in there, but then it gives you an idea of what the inside of one of those granules looks like. And it's not just a bag full of insulin, right? There's a lot going on. And so I thought it would be really interesting for you to really, you're not going to understand all that, but just to be amazed at the complexity of those things and all the pieces, all the proteins that have to be there in order for it to function. And you can imagine how many things could possibly go wrong in diabetes, for instance, that might cause a problem with this system. So let's talk a little bit about the sheer magnitude of those insulin granules. In each beta cell, and there's going to be lots of beta cells in the pancreas. In each beta cell, there are 10,000 insulin granules, okay? 10,000 of these vesicles in each cell. In each granule, then, there are 200,000 insulin molecules, okay? So that means that each beta cell actually has about 2 billion insulin molecules in it. Pretty crazy. So your pancreas makes, and your beta cells make way more, assuming you're not diabetic, make way more insulin than is needed on a daily basis. So it's got a lot of supply waiting to be used. Insulin has a half-life, so it's got a lifespan of very short, like about 4 to 6 minutes. So that allows your body to really regulate your blood glucose on a minute-to-minute basis. So when it's needed, it pumps it out. It doesn't last long, it does its job, and then it gets degraded. And then when more is needed, it's pumped out. So it's a very, very, very fine-tuned system. And so you can imagine how difficult then it is for people who are diabetic for us to be able, for physicians to be able to give patients the right amount of insulin at the right times, because our body does a really good job of it and it's really hard to replicate. Now remember, okay, we go back and we can look at those, that insulin granule picture. You look at the insulin hexamers in there, and you'll notice, so it's not just that they are in hexamers, they are so tightly packed that they have crystals of hexamers. So the hexamers are very tightly packed within there. So it is really, really, you know, like they say, it's crystallized. It's like crystals of insulin. And only after they are released into the bloodstream do those hexamers release from each other, and then the dimers release from the hexamers and the monomers release from the dimers, okay? So that's something that happens only upon release. So how is it that these insulin molecules are kept into these hexamer and crystal formations, these very tightly packed structures inside the insulin granule? I mean, how does that happen? It turns out that the insulin granule has a very special environment to allow that to happen, to encourage that to happen. If you look at the granule picture that is in there, you'll see all kinds of proteins around the membrane. One of the ones that you'll see there is a proton pump. So a proton pump pumps protons or hydrogen ions into the granule. So what would you think would be the effect of, on the granule, of constant pumping of hydrogen ions into the granule? It should lower, it lowers the pH. So the pH on the inside is very low, which means it's very acidic. That, under acidic conditions, the hexamers want to crystallize. The other thing that's there is a zinc transporter. So you should be able to see that zinc is being pumped in as well. Now these hexamers, I didn't really talk about before, is these hexamers are held together by two zincs that are each bound by the B10 histidine of each insulin molecule. So you can see these histidines, three of them come together to bind each of the two zincs. And that's what is required to form a hexamer. So hexamers form when there's high concentrations of insulin, low pH, and lots of zinc, so that they can bind around the zinc. That's what holds them together, okay? When they're released, when the insulin is released to the blood, you don't have the low pH anymore, you don't have high concentrations of zinc, so there's nothing really holding them together anymore, and they want to then come apart. And that's good, because an insulin monomer is what binds to the receptor. This is what is the functional, the active protein. The hexamer will not. So if it's in hexamer form, it won't be functional. And so it's really important then that they're able to break down into their monomeric parts. One interesting anecdote is that that I told you that there's this maturation process. So the insulin granules have to mature before they're ready to be released. But you also make a lot more insulin and granules than you need at any given time. Well, it turns out that once they are mature enough, it turns out that the younger granules are the ones that are utilized first. They're the ones that are exocytosed and released into the bloodstream first. And so you'll have a lot of really old insulin granules that are sitting around not being used for a long time. They're there if they're needed, but the newer ones are the ones that are utilized first. So it's really an interesting process. It's a lot of information. You don't need to understand it at a high level, but it's nice to understand how these beta cells synthesize and process this insulin and ultimately secrete it. We're going to wait for another unit to talk about how that process works, how the beta cells sense high levels of glucose and in turn then release insulin. But this is something that you might want to use as part of your modeling project. Or you might want to do insulin receptor and its effects, things like that. There's a lot of ways you can go now. So like I said earlier, if you're at the point now where you want to start modeling your protein story, you are more than welcome to start working on that. Okay, I think I'm going to stop there. If you have any questions, please use the discussion board and we'll chat about this. All right, nice job. Talk to you later.